So today we continue on how we culture the mammalian cells. The previous class was I give you a principle about the surface protein absorption on surfaces, and mammalian cells recognize it. Also the size comparison between bacteria and also mammalian cells, and media, types of the mammalian cells, those kind of things. So we actually, this is a procedure how we grow the cell. So before we actually start this step-by-step procedure, I have to mention first, we basically purchase small bottles of the mammalian cells that from the so-called purchase source that we call ATCC. Then we have this frozen, it's minus 70 degrees frozen cells. We put it into a culture dish to actually stabilize it. And then stabilize it, and then some number of the cells are die, or most are alive. And then we now have to prepare another dish for another culture. To do that, we have to detach the first layer of the cell and then put it into the another empty culture dish. So that one becomes one dish, it becomes 2 or 3, and 3 becomes 9, 9 becomes 27, for example. So because the cells are dividing and just are growing, so that you have to keep making this initial culture, which is very important, because say we have 9 dishes, for example, start from the 1 and becomes a 9. After one passage and becomes second passage, we actually, some part of the cells, we actually make a store at minus 70 degree for the next, in the experiment in the future. So we don't actually explain this part. We don't explain this part. Rather, the most essential part, the cells are anchorage-dependent cells, so they stick on the surfaces. Then how we detach and then grow the next dish, and then to which they grow again. So this is the procedures that we have to background I explain. So see, all solutions and equipment has to be sterile condition. No bacteria and everything, mycoplasma, everything is just there. Okay, so now remove and discard dispense the culture media. So basically remove the media from the culture vessels. That means I can go back to the previous slide that I explained you before. So now you're sitting down here, and then this is previous culture cell dish, and then you remove this using this pipette, and then you remove these solutions out. You remove and discard. Okay, and put it in here. Okay, so that is the step that I will explain. I'm explaining now. And then they wash the cells using the balanced sodium salt solution without calcium and magnesium, approximately 2 milliliters per 10 square centimeters surface area. So probably I think it's 4 or 5, about 4 milliliters is enough to actually dish, covering the dish about here, and then you slowly wash this. Okay, and then you take the washing solution out from this culture flask, then go to waste. This is the thing. Okay, so gently wash the solution to the side of the vessel, and then you remove it. So then of course you have to remove and discard the wash solution. So this is the basically first step. So you're sitting down, make sure the sterilized all equipment, and then take the salt solution, like we call the PBS, and then you take it. Of course, it's in previous media going out, and then put the washing solution, and you gently stir, and then you take it out again, and then discard. So this is the step. So add the pre-warmed dissociation reagent such as trypsin. So this is the key step, one of the key steps. So trypsin, as I explained before, trypsin and then recognize arginine or lysine and cleave next to it. Remember that in the protein purification part I talk about affinity chromatography, to which we learn about the enterokinase. The enterokinase, the histidine tag, when you talk about the histidine tag, where we attach to the histidine tag of the target. So N-terminus we attach or C-terminus we attach. So I mentioned about the N-terminus is right condition because the enterokinase is recognizing the arginine sequence and then they cleave. Recognizing sequence before, they don't cleave after. So then this trypsin is actually arginine after and then lysine after. So what they cleave, that one of the cells is a membrane protein, and also remember that's why I was emphasizing the serum protein on the surfaces. So surfaces has a layer of the protein to which mammalian cells recognize, right? And then the trypsin comes here. Trypsin goes in the gap, and then the protein has lysine or arginine. So they cleave. Of course, I think they cleave the serum protein. Mostly serum protein absorbance. So they cleave. Anyway, if they cleave, then cells are going up into the solutions, right? But that is after the product of the cleavage reactions of the trypsin. Okay, so that's why pre-warmed is you have to increase the temperature about 37 degrees. Otherwise, the enzymatic reactions doesn't happen. So you have to be the enzyme reactions are temperature dependent. So you have to pre-warm. Dissociation agent means it's a cell dissociation reagent by the trypsin. The thing is, you have to remove the trace of the calcium magnesium, because if you have this calcium, then the trypsin doesn't work. If you go back to your biochemistry textbook, then calcium is interfering in the reaction mechanisms of the trypsin. Okay, so then the size of the plastin use enough reagent to cover the cell layers, approximately 0.5 milliliters per 10. So you put some 1 milliliters of the trypsin, then basically you can T7 dish, culture dish, you can actually cover all the area. And then you have to give it enough time to actually trypsin actually cleaving the protein. So you incubate the culture vessel at room temperature for approximately 2 minutes, or 2-3 minutes, or 5 minutes, depending on what. So note that the actual incubation time actually varies, depending on how many cells and depending on the activity of the trypsin, depending on the temperature, depending on trace amount of the calcium. If you have a very, very small amount of calcium, the activity is just down. So that all depends on the reaction time. So enough, say 3 minutes, 5 minutes, then it's pretty enough. So they observe now cells under the microscope for detachment, because the cells are under the microscope. You can see the bottom of the cell. So now the trypsin working so after all the trypsin is cleaving the cells, and actually you cannot focus on the cell because cells are on the top, so you're out of the focus. The cells are green, like your glasses. You've got borders in here, and then out of focus you can see blurry image. So you can actually see the blurry image without any cells. So it's kind of monitoring the cell detachment by using the microscope. Okay. If the cells are less than 90% detached, increase the incubation time a few more minutes, checking the dissociation every 30 seconds. Okay, this is the things. So the next step is now. This is the after. So now the cell protein, they attach. Trypsin comes in, then trypsin cleave. Cells are detaching from the surfaces. So when more than 90% of the cells are detached, and then you can actually go back to this original, say this trypsin goes in here. Actually, you take this vessels going up, making this up. So because it's only 1 millimeters, so millimeters, very thin liquid layer. So you have to collect it. You have to collect it because all the detached cells are existing in 1 milliliters of trypsin solutions, right? So they have to go into the top, and all the 1 milliliters of the solution going in the corner. So shaking like say going, and then you put like this way in the solutions in here. Okay, and then you collect. Of course, the cells are in the solution. You have to collect that the solutions. Then tilt the vessel. That is what I mean. And then add the two volume equivalent. So if your previous step you have 1 milliliters of the trypsin solution, that means it's twice as 2 milliliters of the volume of pre-warmed complete gross medium. So the complete means the DMM or MEM plus FBS, fetal bovine serum. So that means it's complete. Okay. The reason is if you don't use the incomplete gross medium, which is a MEM or DMM, then the serum the media doesn't have a protein. Right? So without protein means the trypsin. So now the trypsin becomes contaminant of cells, and the trypsin's here. Trypsin is an enzyme that's continuously attacking the cell membrane of the membrane protein of the cells. So as the reactions continues, then anyway they got damaged. Cells got damaged because the proteins are cleaved. It's not good for cells. Cells are getting die. Right? So then now we put complete serum with the proteins there. So many proteins are coming there. Actually, you got a competitive reactions, right? So you got a trypsins here, and then you got so many proteins are by the serum. Then proteins are reacting to incoming the serum proteins from the media instead of the cell membrane proteins. Its number is much higher from supplying from this complete gross media. So maybe times 10 times 6-folded, 8-folded, and then all the reactions goes here. So transfer cells to the 15 milliliters because it's 1 plus 2 milliliters, so about the volume is about 3 milliliters, and transfer the solutions to this conical tube, and then centrifuge about this 200 G for 10 minutes. And then cells are heavy, high density, proteins are still in solution, and then only cells are spitting down. So all the supernatant that you throw that away, right? So then supernatant throw that away, and then resuspend the cell pellet. So the cells are in the bottom, and minimal volume of pre-warmed complete gross medium. Okay, so complete gross medium, and then basically this is it. I mean, you initially got cells attached to surfaces. You basically add the protein allowing the reaction. Cells dissociated, and you got the solution. Of course, you add a complete serum media, and then centrifuge. Cells are spinning down at a pellet. You throw that away. Then you got a new medium is provided, and then you resuspended. The cells are in the solution, resuspended, and then you take the solutions to the next level of the petri dish. And then the cells are going onto the surfaces and anchor, and then they spread it. So this is it. So this is all the procedures. But now is before actually completing this talk, and then this determine the total number of the cells. You have to actually count the cells. How many cells I'm having? Okay. So that is the we called very simple devices. It's a basic glass basis. Some devices I will show you later. Hemocytometer counter and trypambulu exclusion, and then you can actually count the cells. If you don't want to use a trypambulu, I mean trypambulu what does is cells are alive, then they excluded staining of the blue staining. So that means the cells are living. Cells are dead cells, the blue comes stain the cell, so you can actually exclude that cell. But nowadays people don't use it, and people simply just count it, and then because all the procedures very well set up, and then your percentage of the dead cells are very small. Unless you are learning procedure like a very first year graduate students, then you have to use it. and the procedures get used and handled very easily and without any damage in cell, then you get used to it, and then you don't actually need to use these solutions. Okay, so the how to determine the number of the cell is the following. So this is the devices. So basically, you can put it into the microscope, and then you got the objective lens about 10x objective lens, and then you actually see this image. Actually, you see that this image. So now think this is scale. This is 1 millimeters, right? Then this is also 1 millimeters. So it's 1, 2, 3. This is a 1, 2, 3, 4, right? So that means this is the 250 micron, right? So from here to here, 250 micron. So now you can imagine that. How many lines is it? You cannot, because of this resolution, you cannot see it. But maybe you can imagine that this lines are maybe. Oh, this is good. Actually, you can see this area. So this area is this area. You can have a C 1, 2, 3. So divided by 4. So you can see now this is a 250 micron. Actually divided by 4, and then it's about 60 micron. So this, from this one, the pixel unit has about 60 by 60. Remember how the size of the mammalian cells in the beginning of the class, of the last class, about 15 micron, right? Or even larger depending on cells. So that means you can actually see these dots are actually the size of the cells. So you can actually see in these devices of the size of the mammalian cells. Bacteria, 10 times smaller than this. You cannot actually see by the objective lens 10x. Cannot see it. Has to be increased as the objective lens things. So anyway, so this you can actually see the cells. Now you can actually see the counting the cells. Okay. So the region is this is a cover slip. This is a base, and you put this cover slip, and I will do this in this. You put this cover slip, and then you see through this side like this. There's a small gap in between the gap, because this line is the height is a little higher. So this is kind of bridge. You put in here, it doesn't stick onto this area because this is higher. So it's like a little bit more height. Then cover slips in the top, and this area is a little bit lower height. Okay. So then you load cell suspension. So remember that the last step of this, right? So you can resuspend the cell pellet, right, in the media, and then you take a little solutions, maybe 10 microliters or 20 microliters, and then this is look like a triangle. So you can very sharp tip. You actually touch this area, and then there's a capillary force, and you can actually 10 microliters and 20 microliter solutions automatically goes into the side because of this thin. The layers look like this. Solutions are capillary force, and water goes inside automatically. So then this is the volume of this one, the entire volume of this. So 3 millimeters by 3 millimeters. And then you have to think about the gap size, gap height. So if you include X and Y, and there's the Z, you can Z direction, that volume is 10 to the 4th millimeters. The millimeter divided by 10 to the 4th. Okay, so if you have a 10 to the 4th number of this volume, that is one equivalent to 1 milliliters. Okay. Okay, if you have a devices of the say 10,000 devices, then you got 1 milliliters of the solution. You got a graphing over here. Then your 1 millimeters can use it, all of them. Okay, so you cannot, but you don't do it. Just only do one times of this. But anyway, this one part of the 1040 millimeters in here. So basically, you can count it. Count 1, 2, 3, 4, 5, 6, and say count it. So if you have 100 of this, okay, 100 in this area, then 100 of this area, that means you can repeat it by, on average speaking, you repeat it by 10 to the 4th. So 100 here, 100 here, 100 here, 100 here, 100 here, repeating by 10 to the 4th. So the 100 is 10 to the second, 2 power, times 10 to the 4th. 10 to the 4th is 10 to the 6th. It's a 6. 10 to the 6th is a million cells. That's why the total number of the cells is the cells you counted times 10 to the 4. Cells you counted per square and then 10 to the 4. So again, you have 100 of this, okay, 100 of this, then you got a million, because 100 times 10 to the 4th is a million cells per milliliters of the medium that you counted cells. So okay, this one is the actual how they look like. I mean, to which you have cells are at the bottom here, and then it look like this angle. There's an angle because you actually put the pipettes in here, and it actually is easier to actually put the pipette instead of actually put pipette like this. So you can easily put pipette and then take out the media like this, and take out the media, and also you can put the media in here. The reason I bring this to you is actually the number of the cells. Right? This is the this area is about 8 centimeter by 8 centimeters in square. So this is actually 75 square centimeters to which cells can grow. Okay, so this is very important size because there is a reason. Actually, we got this area, not bigger, not smaller. So I will show you what the reason why we have this size. Okay, so then to have this, I mean, this is not a square. I mean, there's a rectangular and plus a little bit different shape. But if you can imagine this area is square, then it's about say 8.5 times 8.5 square centimeters. That means I can put here 8.5 centimeters here. So this is the size about here. And then can now imagine the cells can grow in this area. Cells can grow like this. So remember in the step in this class we talk about the trypsin step, right? Trypsin step, cells detach, and on a cells and attaching here, then they flowing, they dissociated from the surfaces, and then we collected centrifuge. We got a next step, right? So initially, before we have now cells are confluent. We call that confluent. Means that confluent means that cells are without few space, without almost any spaces. Cells are connecting to connect each other. Very high popular density. So cells to cell and cells to cell. There is no space between the cell because they divided. 1 becomes 2, 2 becomes 4. So eventually they become some highly populated situations. So then they populated here. So we can have a very simple calculations that say this 85 millimeters, right? So this is 85 millimeters. So in this is the 85 times 10 to the 3 micrometers. The reason why I changed this is following, because this 85 times 10 to the 3 micrometer in one axis, but remember in the class that how the size of the cells, the size of the cells, for example, is 10 micron. Say it's about 10 micron in one length. So of course, this you have to calculate 2 dimension, but we think about only one directions here. Okay. So then this is about say 8.5 10 to the 3 number of the cells, right? So you can imagine that if you only think about this one dimensions of the cells, you can make an array of the cells about this number, 8.5 10 to the 3 number of the cells. Okay, so this is one direction. So you can have say one directions and one directions here. From here to here, you have 8.5 cell. Just roughly speaking, 8 times 10 to the 3 numbers. From here to here, 8 times 10 to the 3 numbers. So you can confluent situations here. No spaces between the cells. About you have a number of the cell. You can basically roughly calculate it. So it's about 64 times 10 to the 6. So it's about million scale. Million cells growing in here. So it's simple that 10 to the 6. If you have of course, this is the ideal situation. So of course, there is a space between the cell. But when you have this solution, so actually you can imagine that cells are growing here. You put in a microscope. You see the microscope, and then you see everywhere fairly confluent cells everywhere. That means in this here you have 1 million cells. Okay. Actually, this is a 10, but actually bigger than 10. So then this is actually much over than 1 million cells. But actually think about it. I mean, size of the cells is bigger. There's a little spaces between the cells, right? Then it's about million cells existing in here. That's why we have 75 square centimeters. We can calculate it here. It's about 1 million cells. So now, million cells you can grow. Then after million cells you can have this. Then now you have to have it divided. The next step you can bring 10 of this. 1, 2, 3, 4, 5, 6, 7, 10. So that 10 to the 6 of the original dish, 10 to the 5, 10 to the 5th, 10 to the 5th, 10 to the 5th, and 10 dishes you can culture from one here. And then 1 becomes 10, 10 becomes 100. Right? So that is kind of scale, actual scale that we talk about. Right? So this is the first passage, and then the second passage you got 10 dishes of this. You got a 10 dishes starting with a 10 to the 5th, right? Not a 0.1 million cells. Then you only have allowing about, say, to become 1 million, then million cells become confluent in here all over the spaces. So now one division of the second passage becomes 0.2 million cells, the one division. Second division, 0.4 million cells. Third division, 0.8 million cells. So that is now here 3 divisions is possible around. So that's 3 division means you have the cells depending on a cell type, but one cells require 2 days to divide. Then you have a 2 days, 2 days, 2 days, 6 days. It's about a week. Week of the experimental time of the one passage, and next round, then you get at a trypsin that we learn about these procedures that we learn about, say, trypsin and then cell collecting procedure. But next step is actually after about a week, 6 days or a week. Then you have a next experiment after. You have the schedule of this. Then that's why we selected this the area of the space. Okay, thank you very much. This is the end of the mammalian cell culture. And then the next step is we talk about the protein adsorption. So then how we analyze the protein adsorption. Okay, thank you very much.